The part two of this first session is, I think, will be as entertaining as the first two talks of the session. And I hope I get the first speaker's name right. His name is Keolu Fox. And I thought maybe if I can't pronounce the first name, I'd just call him the fantastic Mr. Fox. Or maybe he's the fantastic Dr. Fox, I don't know, probably. I don't know whether you've watched that movie, but I have to say it's one of my all-time favorite movies, but I'm sure he won't refer to it in his talk. The talk has a very provocative title, Guns, Germs, and Alleles, kind of things that we run across in our lives all the time, to a greater or lesser extent, and I'm looking forward to hearing about your view of it.
Thank you. Thanks so much. All right, thank you guys so much for having me. Thank you to all the Larrys. Thank you for really the staff who's been providing us with just incredible food, all the restaurants, even the music that we get to listen to in between sessions is exceptional. So thank you guys so much. I chose this title for this talk, Guns, Germs, and Alleles, and for many of you it might make sense. It's a play on Jared Diamond's famous Pulitzer Prize winning book. And we're going to get into that. I'm going to challenge some of those ideas. And I've chosen some art for you as well here. You can see there's probably no art that comes from back in the past where we actually had deciphered the structure of the double helix. So you can tell that this is AI generated. But it's also an interesting play on like how has our migratory history and our diaspora and our interactions as people, how has that shaped our genomes over time? And then how does that predispose us to disease today? How does that have a role in our predilection to disease? So oftentimes we present the genome as a, sort of a time capsule, and we either talk about the population genetics of something and say, okay, well, we sequenced the genome of a Neanderthal, and so now we understand our past, or that we were intimate with Neanderthals or something like this. But that gets siloed. Our understanding of our deep history gets siloed. Or we'll present something and say, okay, well, here's how we mechanistically understand type 2 diabetes, cancer, etc. But the reality is they empower one another synergistically. So when you take those layers of data and you compare them to one another, you can have some beautiful insights into one, where we come from, but also how that contributes to disease. Okay, so that's a little table setting for today. I'm from Hawaii originally, but I work at UCSD in La Jolla. It's perennially ranked #1 by Surfer Magazine. That's why I chose to work there. And for all you guys that work in like Boston and cold ass places like that, man, hard luck, eh? But it's also a really unique place for genome technology development. Your largest service provider for sequencing, Illumina, is located in La Jolla, California, amongst many, many, many other kind of great thinkers, and it's a lovely place to work and learn and live, except for the housing prices. I am the co-director of the Indigenous Futures Institute for the University of California System. We focus on a lot of things there, but our whole idea is centering indigeneity and understanding the past and how that informs the future. Now, before you say, oh man, here's some millennial talking about some woke bullshit, I want to remind you that this is what Yosemite looked like prior to John Muir rocking up and saying that it looked pristine and beautiful. Okay? It was managed for thousands and thousands of years by indigenous people. Now, when you look at it in 2020, 2022, you can see the density of all of these trees. This is a national park that has not been cultivated and taken care of and loved. And so it is prone to gigafires. And so what we do is we strive to bring together distinct elements of indigenous knowledge systems and distinct elements of contemporary technologies and methods, those two distinct elements, and we recombine them into new alloys. That's what we do, fundamentally, whether it's machine intelligence, remote sensing, architecture, and of course, genome sequencing and editing technologies and the revolution that's happening in precision medicine. And that's my background. And what I focus on is really understanding our diaspora and how we become exceptional. How are people adapted to high elevation? Why am I a cheap date in Boulder? Because I have 2 drinks and I'm already feeling, right? Why do some populations of people thrive at altitude and others don't? Okay? Why do some people have the A blood type and some people have O? There's an evolutionary mechanism and explanation for all of these things. And there are many, many different incredible examples, including the gentleman earlier was talking about malaria and HBV. Well, that's an evolutionary story, let me tell you, about sickle cell and malaria and agriculture, right? That's solid evolutionary science. Then there are many, many others that we're just starting to learn about, like CREBRF, which was recently discovered in Samoa. This is a mutation in this gene that predisposes to obesity but protects against type 2 diabetes. It is also found in 60% of professional Polynesian rugby players. So how we interpret these mutations and what we know about them and what they do and how we narrativize them and who's doing the narrativizing, because we all know that correlative science is not objective, right? We're searching for those relationships. And that's pretty much what I do and what I'm classically trained in as a genome scientist. But we have this kind of dark secret that's going on in our field. And that is that the vast majority of genomes that are sequenced and processed and the insights that we have into human genomics disproportionately feature one community of people, and that is Western Europeans. And when we look at the numbers, these are fresh numbers from Melinda Mills' GWAS diversity monitor, you could see that people who fall into the other or mixed category, that would be me, Native Hawaiians, we have the highest percentage of admixed people in the United States of America by 28%. We are not bearing the fruits of precision medicine. Even though I pay my taxes every year, the future of how we predict, prevent, and participate in disease disproportionately impacts and includes one community of people, okay? So when we look around at a state like California, not to center California because we're in Colorado, but it is the 5th largest economy in the world, 50% of that state is Latino. That is not represented here, right? Now, the far more concerning statistic that you'll see here is when you look at this heat map of the world, is that 72% of these studies take place in 3 countries, right? So we have to think about resources and relationships. Africa, for many of you, this is the most genetically diverse continent on planet Earth. Period. Non-negotiable. Human beings, we've spent the most time in Africa over our diasporic history. Yet, when we look at the infrastructure that's built there, there's nothing. There's very few genome sequencing centers, there's very few data centers, there's very few biobanks, and that means that their genetic history and genealogy gets outsourced to the UK, the United States, and the 3rd largest country is Iceland. When I say largest country, I mean these are the top 3 countries that are doing sequencing. So what does that mean? Doesn't that look like any other research-bound relationship? When we talk about other resources like oil, water, cobalt, conflict minerals. You get the picture. And because of this, there's also a disproportionate amount of intellectual property that's filed. And so here is the dining hall in Illumina's headquarters, and you can see all of these successful patents that have been filed that are on the wall here. And to me, it resembles like a hunting lodge where you have a bunch of moose heads. But when we look at the numbers, again, 45,000 patents have been filed worldwide that include DNA sequencing, and that was before the pandemic. So there's this windfall of intellectual property and resources that are generated in this space, and a lot of times we can derive new and novel mechanisms from indigenous people's genomes, for example. Like high elevation adaptation might kind of serendipitously lead to the next Viagra. That's a multi-billion-dollar drug that a lot of people use, right? However, proceeds from the development of that drug should go to the people that enabled it to be possible. So that's another thing we work on, and we call that benefit sharing. And I'll get to that in a moment. But this relationship is not equitable. So when we say DEI, some of us mean diversifying my equity and investments. And that's why we started the Native Biodata Consortia. This is the first genome sequencing center and biorepository on an indigenous or Native American reservation in US history. Here are my partners. And we're doing all kinds of things and projects, and a lot of this talk isn't about that, but I encourage you guys to check it out. That's why I wore the sweatshirt here. Now, I'm originally from the Big Island. This is Kohala. This is where my ancestors are from. We now live a little bit south of here. Uncle Larry lives on the other side in Kona. But it's a really beautiful and diverse place. It is one of the most biodiverse places in the world. We have the most diverse soil regiments in the world. From the top of Mauna Kea to the bottom of the ocean is the largest geographical feature on planet Earth. It's a scientific epicenter for volcanology, astrophysics, geology, marine biology, and many, many, many other things. It is also the extinction capital of the world and the invasive species capital of the world. So when you look at those IUCN red lists, Hawaii is always the place where you're seeing the highest level of extinction and invasive species. And by invasive species, I don't mean tourists. So we, people who are from there, are acutely sensitive to all of those systems and those dynamics and that biology and the tourism industrial complex and the military industrial complex and colonialism and many things that are facts. And it's also the most diverse state in America by a country-ass mile. The county I'm from is the most diverse county in America. It's not Queens, it's not Houston, it's Hawaii County. That's followed by Maui County, Kauai, and then Honolulu is actually number 11. And the people that are there have very unique genomes. Again, extremely admixed. We produce, I don't know how many professional athletes. It's unbelievable. But we also have health disparities that exist within our communities as well. And understanding the genomes of the people that live there is extremely complicated because we're dealing with of the most complicated levels of mixing. And because of that, there are lots of different conversations about what happened initially. First, we have the Polynesians who arrive on boats about 1,200 years ago from Tahiti or Marquesas. It's not clear, but we'll get there. But the way it's narrativized often by people like Jared Diamond is that, oh, it's guns, germs, and steel. The West has dominated and has had this inclination for colonialism for these reasons. But I'm here to tell you that it's more like germs, germs, and germs. And the central idea that we're going to talk about is that when Cook or Cortez or any of these individuals arrive, they bring with them all kinds of things. Yes, they bring technologies like metal, but they also bring things like syphilis. And that has shaped the contemporary genomes of indigenous people today. And now we have the technologies to identify those things at a high resolution using genome sequencing technology. So I'm going to tell you about how we're doing that. I'm going to break down a few different examples. But the central point here is that the way the conquest of Mexico and the Aztecs from Hernan Cortez, for example, is represented, that they came through, conquistadors in shining armor on horses, etc. But the reality is the totality of actual deaths because of battle and war was probably around 200,000 people. The number of people who died because of smallpox is probably around 20 million dead bodies. And that population collapse shapes the genomes of contemporary people today, and we finally can detect those things, okay? So bear with me. That's the conversation. In population genetics, we refer to these as bottlenecks. Who's ever heard of that term before? Okay. In contemporary terms, that could also be like understood as genocide. But we narrativize the term as a population bottleneck. And what's happening here, and you can see, is we have an original population, we have our bottleneck event, and then we have a surviving population, okay? We then have time. We have the introduction of a pathogen like smallpox, syphilis, or leprosy. And then we have a reduction in something called HLA. Who's heard of HLA? The human leukocyte antigen. Okay, very cool. This is a really, really important mechanism for thinking about and how we manage and attenuate inflammation, the root cause of all common complex disease. So it's very important that we understand it, especially when we know that 95% of clinical trials exclusively feature white people. So we have all of these variables going on in the background, and we're not really designing drugs with precision, even though we call it precision medicine. Does that make sense? Okay. Okay, so for the rest of this talk, I'm going to talk to you about 3 stories that center the Pacific. So we're going to take a trip. First, we'll talk about the paths and timing of the people of Polynesia. How did this happen? How do we know what those timings are? What are the pathways in the largest ocean, the most ancient ocean on planet Earth? You know, how did that go down? And what lines of evidence do we use to understand that? Then we'll talk about the origins of leprosy or Hansen's disease in Hawaii, in Oceania, and I'll talk to you about how we use genome sequencing to understand ancient leprosy strains, working with our oldest hospital from 1859 in Hawaii. And then finally, we will talk about the largest study of its kind that we just completed in French Polynesia. And this is a look at gout, which is not a popular thing to talk about, but it is the levels and the reason why people are calling this an epidemic, and you shouldn't loosely throw around epidemic and pandemic these days. But I think we conclusively showed that the rates of gout that we see in French Polynesia are much, much, much higher than expected. Okay? All right, so first, who's been to Hawaii before? Nice. Where'd you stay, the Hilton? Mm-hmm. You took a plane. My ancestors did not do that. They took different boats, catamarans, double-hulled canoes that were developed over time, iteratively. This is what Master Navigator Nainoa Thompson refers to as the original moonshot. And so using the celestial sphere, understanding that stars pass overhead, understanding bird migration patterns, the green glint on the bottom of a cloud reflecting from the bottom of a lagoon, all of these lines of evidence, this scientific method, that's how we got there, okay? And I've always, even when I was a kid, I've been obsessed with where do these time estimates come from? Like my grandma would say, oh, we came from Nukuhiva Marquesas. And I'd be like, okay, but how do we know that? How have we reconstructed this? How do we know that it's, 2,000 years ago or whatever? And there are many ways that people do that, right? There's archaeology. Famously, here are the Moai and Rapa Nui. Sometimes you have these really elegant and beautiful applied algorithm approaches or simulations, right? People have been doing this since the 70s and saying, okay, what's the probability if it's this far away, or if the calculus that you need to understand different celestial patterns and constellations is different in the northern hemisphere, then doesn't that mean that you basically trained a whole system on the southern hemisphere? The probability would be lower to find a Hawaii versus a Tahiti or something like this. So they had some really kind of cool mathematical models where they were looking at that. Linguistics. Some of the words we say are ubiquitous in Oceania, like Moana. Who's seen that movie? You guys seen that movie? That's pretty ubiquitous. In Hawaii, we say Moana, it means the ocean. In Tahiti, they say Moana, it means the ocean. In Aotearoa or New Zealand, they say Moana, it means the ocean. Manu means bird. But once we break down other words, aloha, talofa, aroha, linguistics, we understand that. Very important way to distinguish between Austronesian cultures and communities. But my favorite, and the reason I'm talking to you today, is that we can also use the human genome to decipher that and understand our origins using population genetics. And we know how some of these interactions started. Here's the arrival of Cook's boat. There are all kinds of historical manifests. This is what one of these interactions might have looked like. And we know how it ended. Who's from England? My guy's from England. You guys know how it ended. We ate that dude. And that's represented in certain ways historically, and we use archival information, and we often layer all of those different types of information to understand what's going on. Now, Cook is celebrated as one of the greatest navigators in human history. He came to Polynesia for a reason, and that was to observe the transit of Venus. It was a very, very important collection of scientific data at that time, right? You could think about it like the ancient version of launching the James Webb Telescope or something like that, right? But what you don't know is that his golden goose was this man, Tupaia. This is the actual greatest navigator in human history. You have been brainwashed to think that Cook was good at his job, and he was. He was a great cartographer. He was not a high-born elite, person. But the reality is that this man was a master navigator, Tupaia. And he knew and had memorized the locations of over 1,000 different islands in Hawaii, in Australia, you get the picture, right? So he basically golden goosed him, and he used him to identify and find all of these different places. Now, Tupaia was like a true, brilliant imagine, you've never used a paintbrush before, and you're creating art like this. Now, one thing that came out of their interactions is this map. They came together in the HMS Endeavour Resolution. I forget which boat it was that they were using at the time. And they created this map. Now, this is a true collaboration between two different masters of their fields. And you can imagine them pouring over whiskey in some, cabin. And this is a way to view the world, but this is not how Tupaia viewed the world. This is him translating his knowledge system into the way that we view Polynesia. And it's truly remarkable because it demonstrates that there are ways to collaborate with people that are not from your communities, but bringing attention to the fact that Tupaia was actually the A side in this relationship is one thing that I wanted to do here today. But it brings up a bunch of scientific questions that I think are really important. And it's, who's in control of the narrative? So up to that point, nobody in this room had heard of Tupaia. Maybe Larry did. But you thought he was the greatest navigator in history. And basically, Cook took credit for his work. So who's in control of the narrative? And how do we recover and make sure that those things are accurately represented in human history? And then who's biasing the data? And then whose hypotheses are we testing, right? And ultimately, is it even your question to ask? And that's an uncomfortable question for a lot of people. And I think it's really, really important. And I put a picture of Thor Heyerdahl up here. Who's ever heard of Thor Heyerdahl? Okay, yeah, Thor Heyerdahl, Kon-Tiki. He set out to demonstrate in the 40s that Polynesia was settled the opposite direction. What we know to just completely not be true. We know a lot of things now. We know that our migration out of Africa bifurcates into a number of different directions. We know that we move into Asia, into Taiwan, deeper into the Bismarck Archipelago, and then further on into these original moonshots, deep into the Pacific. This version is not true. We didn't come out of South America and then go in the opposite direction. Our ancestors did make it to South America well before any, Western European person did, and there's a whole bunch of lines of evidence that have shown that, including the human genome. But one thing that Thor Heyerdahl did is he set out and it's the birth of something that we call experimental archaeology. And so he built a boat that he was going to float over, and it didn't work. It didn't mean that it wasn't a good idea, like the idea that you should test your hypotheses with functional evidence, right? Especially now more than ever, all these AI people. Man, AI is the new plastic. We know that, right? But you have to create functional lines of evidence. And this was the birth of something really beautiful. And this, while it was unsuccessful, it inspired the Hokulea. It inspired a rogue bunch of anthropologists and native Hawaiians to build a double-hulled canoe and sail it from Hawaii to Tahiti to demonstrate that that's where we came from. And this resulted in a renaissance in our culture at a time where it was illegal to speak our language, where you would believe Thor Heyerdahl over these master navigators and sailors who spent every waking moment of their life on the ocean. Maybe you should ask those people where they came from. So, and look at that party. That looks like a good party, eh? All right. But in Hawaii, we always say, "Ika wa mamua ka wa mahope," and this means walking backwards into the future, asking your ancestors for advice, acknowledging the mistakes you've made. And so for us to be able to use human genome sequencing data from many different communities in which we work and create partnerships to understand our diaspora and create a higher resolution look of that and publish it in Nature, and the gold standard, as you all know, and get to use our language as descriptors in the maps that we create feels really, really good, okay? I'm going up for tenure this year. Feels really good. But that doesn't mean that the media is going to get it right? This is what Science Magazine said about the piece that we published. We worked for 8 years on this piece. And they said, no one could have predicted. And they used this old-ass photo. It's like, we know America's Cup and all of those boats are derived after our intellectual property. They just use carbon fiber and hydrofoils. Why you got to use this old-ass boat? You know. But, and in typical fashion, if you want something done right, you have to do it yourself. So I wrote this piece in Scientific American. This is our boat that I learned to sail on as a kid. This is Kiakahi. This is in Keaukaha on the east side. And we got to speculate and summarize and talk about this article for the next generation of people. So it was pretty cool, pretty sweet to do that. Okay. I see that I don't have that much time, so I'm not going to be able to finish all these stories. So I will rapid fire through this next one. But one of those things, again, that was brought over in those interactions between, let's say, Cook and some of our ancestors were different diseases. And one of those famously that ends up on the shores of Hawaii and impacted our community greatly was Hansen's disease, leprosy. And so here's a pretty cool timeline and demographic history of that from, it's a collaboration between Kamehameha Schools and the Pew Research Institute. And you can see here we have the number of people in the thousands, and then we have the time estimate. So this is the arrival of Captain Cook. This is the identification of the first case of leprosy, and you can see like steadily the population is dropping off. And then you can see the establishment of Kalaupapa. So this is the largest leper colony or Hansen's disease colony in the history of the United States. It is also the largest case of medical stigmatization in the US history. And it was pretty gnarly. It is located in an area. Can I play my video? How do I play it? Let me see. Is it working? Oh, can you guys play this for me? Because I want you guys to see it. It's a really beautiful place. But it is, and I hope it works, this is what Robert Louis Stevenson referred to as the pit of hell, a prison fortified by nature. It has the highest sea cliffs in the world. And what they did is they built a natural leprosarium into the mountains of this place. And it was pretty crazy. And forever, and including to this day, people with Hansen's disease exist there and thrive there and live there. That wasn't always the case. We wanted to know how leprosy got to Hawaii and where it came from. That's like a basic population genetics question. But in order to do that, you have to dig into the past. You have to have access to ancient strains of leprosy itself, the bacteria, the rod-shaped bacteria itself. And it's not like we're operating in places where this type of science isn't culturally sensitive. It's like the highest level of community engagement, partnership, and participation to prioritize these questions, even as a Hawaiian person. And in the background, there are all of these other things going on, including major protests around the construction of new telescopes in Hawaii. So I'm bringing that to your attention just to say that these are some of the most culturally sticky and complicated places to work on the planet if you do things the right way. If you don't just take, if you build consensus and you iterate and you allow community engagement to guide your practice. And that's precisely what we did. And we also know that there still exists around over 200,000 cases of leprosy every year. And that there's no vaccine. There are multiple types of antibiotics that you can give to people that work well, but it's still a neglected tropical disease, and most neglected tropical diseases are neglected because there's no access to healthcare. So we're working in a lot of different places like that. We know that there's 2 different types of leprosy, and forgive me, some of these pictures are, a little gruesome. And sequencing those different strains and understanding and classifying them is very important. Taking this population genetics approach and understanding what's the size of the genome of this bug? Where does it fit in relationship? Oh, okay, isn't tuberculosis its closest cousin? It also is involved in zoonotic transfer, so other creatures can acquire leprosy like armadillos, which are very, very, very interesting. And we also know that there have been multiple strains that have discovered that are antibiotic resistant. So we work with collaborators in American Samoa, and they've shown that there are strains of leprosy that are antibiotic resistant, which is pretty terrifying. We know that the genome is around 3.3 million base pairs. It's rod-shaped. And we also know that this isn't just a chance for us to create more equitable relationships in terms of how we think about neglected tropical disease, but there's also an unintended consequence, and that lepers, or people with Hansen's disease, have really, really low rates of cancer. So there's something going on here. There's a chance to understand cell turnover and others, and people and investigators at the University of, I think it's Edinburgh or Edinburgh University, I've never even been there, forgive me. But they're working on this new mechanistic question. So there's more value in sequencing ancient strains of leprosy than just thinking about it in context of where it came from, is what I'm here to tell you. And we got to work with our oldest hospital in Hawaii since 1859. It was established 30 years before the NIH. And there were different biopsy slides that have been banked there. And so we took those old biopsy slides. And if you're familiar, this is kind of the process of how you get paraffin-embedded biopsies and turn them into slides and do like basic pathological workups. And what we did is we moved them into an ancient genomics workflow at Arizona State University. We first identified the strain, then we began to reconstruct ancient leprosy strains that we had access to in that hospital. And we identified a number of new strains that had never been seen before. We published that work in the royal proceedings, and this work is still all ongoing. And we still don't know where it came from. But hopefully we will in the future. So it's pretty cool story. But the reason why I'm centering that is because things like leprosy have shaped the genomes of our communities today. And here's another example of one of those population collapses in British Columbia in the Metkatla community. And what they showed is that by looking at ancient genomes from that community and comparing them to modern ones, there's a reduction in HLA diversity, which is the exact same thing that we're hunting for. And we also know that S.K. Villerslev's group just published 4 papers showing that the population of history of Europe is associated with modern day susceptibility to MS and Alzheimer's. So this is a moment in population genetics where we can understand our diaspora and how it's impacting disease susceptibility today. And that will have clear impact in the types of drugs and treatment that we develop. And so understanding our history is more important than ever. One thing we learned in the pandemic is that Pacific Islander populations were really hard hit by the COVID virus. And that it's hard to say if it is innately genetic or if it's that Polynesian people and Pacific Islanders live in houses where they're multi-generational and you have lots of different people coming in and out. And, you know, it's a really, really hard question to approach and ask and break down. But we do know that there's this reduction in HLA diversity, and that has a direct role in inflammation, and inflammation is a really, really important way that we can think about COVID susceptibility. Uh-oh. All right, I'm running out of time. But our last story brings us to Tahiti. And this is the site of the Olympics this year, just so you guys know. It's going to be really beautiful. For this project, we got to work with the Vice President's office. And what we've learned and found out, essentially, some of the work we've published, we worked with Variant Bio to do this. We've established benefit sharing models there. We're giving free access to any sort of drug or therapeutic that we create. But what we've learned, and we got to use some incredible point-of-care technology tools, is that gout is estimated to affect 1 in 7 people in French Polynesia, which is crazy. And there's a direct relationship between how we attenuate inflammation, HLA diversity, and why gout levels are so high. So this is ongoing work, and we think that we have an opportunity to not only understand how our diaspora and colonialism has shaped our genomes and our susceptibility to disease, but build new drugs in partnership with our company, Variant Bio, and the French Polynesian government. So, okay, I don't have that much time, but I think you guys get the picture. And finally, I just wanted to give you a little public service announcement about how bad AI and machine intelligence is for the environment. It is not independent of the second law of thermodynamics. GPUs are bad, okay? They're good, but they're bad. They require a tremendous amount of energy and information. And there's a chance that heat emissions from data centers could surpass the fossil fuel industry as the #1 contributor to the climate crisis in our lifetimes. And I think that's something that everyone needs to consider greatly. And here are a whole bunch of articles about that stuff. And finally, that we will be having a whole session on Earth-friendly computation at the Pacific Symposium for Biocomputing in Kohala in January. And if you'd like to submit an abstract or submission, please do. And the website is below. And I will do my best to take some questions. Thank you. 4 minutes.
Thank you. Yeah, you've got a few minutes. We've got time for 3 or 4 questions probably. Let's see what we've got. It's difficult to see where the hands are with the lights in the way. Oh, well, one Larry question to start then.
There were so many startling things, but something that you said early shocked me, and I may have misunderstood it. You said, I think, that the rate of species extinctions was higher in Hawaii than anywhere else. I think you said that, and I would have guessed the opposite.
Right.
So would you explain a little, did I hear it right, firstly? And if I did, why is that? That's so weird.
It is weird. First of all, thank you for having me. Okay. Second of all, we're working with data that we have collected, I'm sure. I think one thing that we know about tropical environments and island ecosystems, trophic systems, and island ecology is that they're extremely fragile. And so I think that plays a large role in terms of like how we think about extinction, the rates of extinction, and the impact of invasive species. And so I think there's a direct relationship between how much extinction we see and then how much invasive species we have. I mean, look at Lahaina. How did that happen? Well, we have the most deadly fire in US history because we've introduced invasive grasses from the Serengeti. So there's kind of this unfortunate relationship between those two things. But yeah, you heard me right.
So
So I was interested to hear that people with leprosy are protected against cancer, because so are people with Down syndrome.
Really?
Yes, and I've wondered, especially certain cancers, so we should compare which ones, but I've wondered whether maybe the people with Down syndrome at least get cancer, but the cancer can't grow. So they can't get a blood supply sufficient to allow the cancer to develop. And I wondered if you had any thoughts about why people with leprosy are protected.
Yeah, that's really interesting. I had no idea about that, about the, here's what we know. All of that stuff coming out of the University of Edinburgh is brand new. And what they've found, so first of all, they have an armadillo farm. And they are, in controlled ways, I think, exposing armadillos and cells to the bacteria. And then they're observing different patterns. And one thing they know for certain is that it arrests the development of liver cancers. So that's, so instead of extrapolating this to all cancers, but yeah, there is a direct relationship there, and it's something to focus on. But I think the most interesting thing about that is, you know, we spend so much time and energy and sequencing might and compute on understanding common complex diseases, and sometimes it's just good to change our angle and perspective and say, okay, what can we learn about a neglected tropical disease, and how does that impact other things? What are the unintended consequences of this? So I just think it's a clever, keep your eyes peeled on that though. Yeah.
Cool, thank you very much. We must move on to the next talk. Thanks so much
Thanks,
for
guys.
that.